Hi, welcome to the After Cafe presentation on studying biological and soft materials in their native hydrated state. My name is Sarah Keemley. So, why study samples in their hydrated state? To look at or examine the native structure, to study movement or dynamics, to look at growth, and also to examine mechanics in the native environment. So what are the different ways to examine hydrated material at the MCL? We have optical microscopy, liquid atomic force microscopy, environmental scanning electron microscopy, liquid transmission electron microscopy, Raman spectroscopy, and X-ray scattering. If you look at the image on the right, you can see a stereo microscope image examining microdefects. So when I was a postdoc, my PI always said, did you just look at the changes of your surface? So sometimes it's just as powerful as taking a low magnification image of the material being examined. But optical microscopy is not just looking at things. So this is an image of a flower, and they use different stains and labels just to identify different components. So you can use lectins or fluorescent labels to highlight different regions, and that can help you enhance the contrast. While using optical microscopy. Another form of microscopy that can study hydrated samples is atomic force microscopy, also known as scanning probe microscopy. It's a surface scanning technique that has sub-nanometer scale resolution. It describes a group of techniques used for non-destructive surface studies at the nanoscale. They have a resolution in the order of 10 to the third times better than optical microscopy resolution limit. It's widely used to collect data on various mechanical, functional, electrical properties at the nanoscale, as well as topography or surface study. So what applications of AFM work in fluid? There is high resolution imaging in fluid in the hydrated state. There's nanomechanical mapping, nanoindentation, high speed AFM to monitor molecular movements, and you can also functionalize the tip to examine the molecular interaction. All right, so let's first talk about the high resolution imaging in the fluid and hydrated state. So AFM allows the observation of the surface topography without any sample manipulation, such as staining, labeling, or fixing. The main advantage of this technique is the possibility to operate with a high resolution in air or in fluid in the real time and the nanometer scale. So first we're going to look at a AFM height maps of the SARS-CoV-2 virus. And in the center is a study of prenatal embryonic mouse skulls and looking at bone formation. And on the right, there is the cellulose microfibrils of an onion epidermis. So here's another example of high-resolution AFM scanning. This work was done by Michelle Fromell. She used chemical patterning with digital light projection polymer brushes to do photolithography. So they put the chemical pattern down and then use a wavelength of fluorescent light to cause the polymer to grow. And then they did AFM scanning of the surface and was able to see a growth of roughly 125 nanometers of growth. So this study used a combination of AFM and fluorescent microscopy. So that is also called correlative microscopy. Another application of AFM in fluid is nanoindentation to look at the mechanical properties of your surface. So here's an example from the Cosgrove group done by Liza Wilson and myself. So for nanoindentation, you're doing a force distance curve, and that allows you to look at both the slope of the line and the adhesion of the tip to the sample. So in this case, we looked at the physical conditions of the pectins in Muro. And the question was, do pectin matrix in the cell wall soften as temperature increases, similar to isolated pectin gels soften? So here's an example of an onion epidermis. We used the heating stage on the AFM. The onion epidermal peel was glued down on a Petri plate. The Petri plate was filled with different buffer, and then the AFM head was dropped into the liquid. And here are the force distance curves for this study, or examples of the force distance curves. So these are 3 examples out of 150 curves taken. So we looked at the samples at 22 degrees all the way up to 60 degrees. And this shows that there was no statistical difference in the softening of the pectin matrix in the onion epidermal cell walls by heating. So we found that there was no mechanical effect. Another example of applications for the atomic force microscope is nanomechanical mapping. For nanomechanical mapping, you don't only get the height or the topography, you also get your mechanical channels. You get modulus, adhesion, indentation, deformation, and many more. So here's an example from the Richmeyer lab, mostly done by Julian Guilford Mahoney. So you have a embryonic mouse skull, and they labeled with a fluorescent tag to label the bone, developing bone. And so we were able to probe the different regions of this bone. So in this case, they're landing on the cartilage and looking for developmental changes in the cartilage at different days of development. So here you have the different AFM topography images of looking at the different kind of topography of the cartilage. So here's an example of the nanomechanical maps. We have the height map, and you can see webbing of the surface, so of the developing cartilage. Here's a peak force error that helps you look at the 3-dimensionality with height changes. We have the modulus map and also the adhesion. So we found that the modulus ranged from 200 to 800 kilopascal with an average of 408 kilopascal across the entire image. So if you were to do one measurement, you might get 408, but being able to actually look at the map and of the modulus allows you to try to figure out, like, what is the stiffest part? What part is actually carrying the force? So here's another example from the same study, looking at the location of developing bone. So again, we use fluorescent label to find the different locations, and then scanned it, and then set the modulus numbers, determine the modulus numbers, and we found that there was a increase of the modulus, or the stiffening, along the developing bone. Another application for AFM in liquid is using a functionalized tip to examine molecular interaction. So for this case, you bind a ligand or protein or a molecule onto the end of an AFM probe, and then you're able to examine the binding and unbinding mechanics of the molecule or ligand to the surface. So here's an example where they examined ligand binding events on bacteria trimers and also cartwheel-like structures. And so they looked for both mechanical and topographic changes for the binding and unbinding of the probe with a ligand attached. Another type of AFM scanning is high-speed video frame rate AFM. And so this is a very fast AFM to look at molecular movements or growth. So in this case, they looked at the crystallization patterns of molybdenum sulfide binding peptides, and they're able to watch the growth of this material under the AFM as they were scanning. So at the MCL, we have 3 different types of AFMs that all work in fluid. We have a high-speed variable environmental AFM from Asylum Research called the CIPHER. We also have a correlative microscope with AFM and fluorescence called the Bruker Bioscope Resolve. Also, the Bruker Dimension Icon, which is the workhorse of the AFM, is also able to function in fluid. So next up, we're going to talk about environmental scanning electron microscopy. So the E-stem, as it's also called, expands the boundary of the traditional scanning electron microscope to deliver deeper insights into all types of samples. E-stem allows imaging of samples with minimal preparation and adds variables such as hydration, thermal cycling, and an introduction of gas to characterize in situ dynamic changes. Using water vapor and temperature control stage, some of the most impossible to image samples, such as dirty, highly outgassing, naturally hydrated samples whose properties will change upon drying, are able to be imaged. With full control of the sample hydration, materials and life science scientists are able to observe real-time material interactions in solutions, how humidity changes a sample, how water interacts with the surface of the sample, and moves the research possibilities from pure static points in time to a dynamic and in situ experimentation. But the basic difference between environmental scanning electron microscopy and typical SEM is that the sample chamber of the E-stem can contain certain amounts of gas, most likely water vapor, whereas the pressure of the gas close to the electron source of the microscope remains sufficiently low. So if you want to ask about E-stem, you need to contact Julie Anderson. So here are two examples of beautiful E-stem images. There is an image of the biofilm created by the diatom Didymosphenia geminata, and so these scientists were looking at the biofilm development of this diatom. And here is a very cool example is looking at the water droplets on the surface of hydrophobic paper. So at the MCL, we have a Quanta 250 that has the E-stem mode using a cold stage and high humidity. So the E-stem technology allows for dynamic in situ imaging. Using a Peltier stage can be used to control the temperature of a small bulk sample between -20 and 50 C. The humidity can be adjusted in the chamber from high vacuum to 100% relative humidity. It allows for the study of samples at different percentages of relative humidity. So here is an amazing image of a pollen sample with 100% relative humidity, formation of water droplets on the surface of concrete. And here, this is a wet mammalian tissue looking at the hydration. Again, if you're interested in E-stem, please contact Julie Anderson. Another example of different types of imaging for liquids or hydrated samples is liquid transmission electron microscopy. In this case, you take a liquid sample and you seal it in different windows and then insert it into the TEM. And so in this example, they are able to look at non-pathogenic human parvovirus and able to get a time series of the movement of this virus in the TEM. So if you're interested in liquid TEM, please contact Jen Gray. Another example of liquid TEM is in this study, they looked at the nanocrystalline movement. So they were both using the beam of electrons, they were able to actually write and erase gold nanocrystals. And so it's different time series of the spreading of the nanocrystals and the aggregation of the nanocrystals, a different time series. Another way to examine hydrated samples is using Raman spectroscopy. So Raman spectroscopy examines vibrational spectroscopy technique, which provides information about composition, orientation, and strain. It does not require major sample preparation, and therefore you're able to do a rapid assessment of samples. So IR spectroscopy and Raman spectroscopy are complementary. Water is an IR absorbing material, which limits the ability to study wet samples, but water has a very weak Raman absorption, so we can use Raman to study wet materials or cells in growth media. So here's an example from Dan Cosgrove's lab done by Jingyi Yu and Max, where they looked at the Raman spectra across the junction regions of onion epidermis. And they looked for both the pectin peaks and the cellulose peaks across this junction region. And if you're interested in Raman spectroscopy, please contact Max Wetherington. Here's another example of examining hydrated samples with the Raman microscope. So they looked at identifying local atomic environments of the copper state in alkaline electrochemical systems. So this paper was published in this year from the JANIC lab. So the measurements were performed in the electrolyte rich environment, so they could simultaneously run cyclic volumetric measurements while recording Raman, looking for voltage potentials across the electrolytes. So they were able to simultaneously examine the location across the electrolytes and also the changes in the Raman spectra. So this experiment was done with simultaneous Raman and fluorescence recovery after photobleaching. So use a confocal microscope with Raman capabilities to look at the movement or the correlative characterization technique based on fluorescence measurements in a Raman spectroscopy setup to study the lipid structure and the dynamics of an epitaxial graphene. So this paper was published from the Robinson lab in 2019. Another way to study hydrated samples at the MCL is with X-ray scattering. The MCL X-ray scattering facility has a dome sample holder that can be used to measure the air-sensitive samples or to maintain a humid environment during measurements. In addition to that, there is a humidity chamber on a different instrument, which consists of two components, a humidity generator and a sample holder for multiple samples. The stage is used in the transmission geometry, and the stage can be operated from 3 to 90% humidity from temperatures between 25 and 60 degrees C. So here's an example of looking at hydrated material with X-ray scattering. So in this case, they were looking at preventing and minimizing alkaline silica interactions in concrete. So the alkaline silica interaction occurs when silica in aggregates dissolve in an alkaline concrete porous solution and produces an expansive gel that can cause concrete to crack or fail. So this was done by Tiffany Seisel in the Raj Kapoor lab in PSU Civil and Environmental Engineering. So in this study, they looked at the constant moisture interaction over a 7-day period. And so they did multiple electrolyte scattering spectras to look for changes in the composition of the concrete sample. And they were able to look for the consumption of the unhydrated cement in different phases and look for the formation of Portlandite to occur. Another example of examining hydrated samples with the X-ray scattering is examining a Arabidopsis stem in a humidity chamber for small angle X-ray scattering and wide angle X-ray scattering. This work was done by Josh Del Mundo in Enrique Gomez's lab in chemical engineering and material science and engineering. In this study, they examined Arabidopsis stem bases using wide-angle X-ray scattering technique under relative humidity levels that varied from 2%, 2.5% to 95%. And using the Zeus humidity chamber, they were able to look at the changes in the cellulose crystallinity in changes from the hydration state. So they found that both reflections of the cellulose crystallinity were more intense and broadening with decreasing humidity, making them appear to combine into one peak for higher intensity values. The water is altering the background of the other cellular wall components to cause the 1-1-0 and the 1-1-0 region to increase in intensity, making the two peaks less discernible from each other. At the Material Characterization Lab, we have lots of different techniques to examine hydrated materials, including optical microscopy, atomic force microscopy, environmental scanning, electron microscopy, liquid transmission electron microscopy, Raman spectroscopy, and X-ray scattering. So these are all the instruments that are able to examine hydrated sample, and these are the people that you need to contact to get more information. I hope you got value out of today's talk. Thank you for coming. Bye.